Okay, welcome to Unit 5 Lecture Video 4. This is going to be a short one. It's only like 4 slides, but we're going to talk about DNA mutations. So we're talking about DNA mutations, not chromosomal mutations. Next chapter, we're going to talk a little bit more about chromosomal mutations, which are a little bit different and generally cause a much bigger problem. So mutations are any change in the genetic code. It doesn't matter. It can be, they can have 3 different effects. They can be beneficial, lactose tolerance. So lactose tolerance, if you are lactose intolerant as an adult, okay, or as at your age, because you're technically medically considered an adult, that's normal. We aren't supposed to process milk anymore. However, in a very small population in Europe, and they've studied this and they've seen what's called an evolutionary sweep, because what's happened is over the course of time, or over the course of the winter, they couldn't farm. So babies that couldn't process lactose didn't survive over the winter, and babies that could continue to process lactose continued to survive. They were able to pass on that mutation, and now it's pretty universal in many populations. If you are an ethnic heritage that comes around the equator, for the most part, you might still be lactose intolerant because they could farm, so they didn't need that. And so it's very distinct where that mutation has come from, and they've mapped it out. It can be harmful, similar to what you saw in the video with cracking the code, cystic fibrosis, Tay-Sachs disease, or it can be neutral. And blue eyes, we're not entirely sure how neutral that is. It may have conferred some night vision benefits, but for the most part, blue eyes doesn't really do much from a survival standpoint. So there are 5 basic mutations in our genetic code, depending on what when we're talking about the DNA. So substitution mutations is where we actually switch out a nucleotide. Silent, so that means we change one nucleotide, but it doesn't actually change the amino acid. That wobble position, okay, that we talked about in the last lecture, is because that third base could be a few different bases. And so if we change that third base, a lot of times it might not actually affect the amino acid at all. Missense, so that's where we change the amino acid, or we change the nucleotide and it changes the amino acid. And then nonsense changes the nucleotide and turns it into a stop codon. So that is where it actually shortens up the DNA strand, or technically it'll shorten up the RNA strand. It'll shorten up the RNA strand a lot. And then we have frameshift mutations where we insert or delete nucleotides and we basically change the whole reading frame. Because remember, 3 nucleotides, okay, starting with AUG is our reading frame, and then moving on. So every 3 nucleotides is our reading frame. So if we put in a nucleotide, take out a nucleotide, it changes the whole strand. And then anytime we talk about one nucleotide, we're talking about a point mutation. Now, frame shifts, we can also insert more or less depending on what we're talking about, but we'll just focus on one for right now. So here you are. So we've got our normal strand here, okay? And then if you look, that was where our mutation was. So there was no effect here. Okay, so that's a silent mutation. Silent. Here, okay, we changed it out and we ended up changing the amino acid that goes in there. Depending on what amino acid we change, could have a varying level of effect. Okay, this amino acid, glycine, could be very similar to alanine. Okay, could be very different. So it also depends on where in the protein it shows up. So these can have a variety of effects. Okay, nonsense. So here you see we went from 4 amino acids in a string, 1, 2, 3, 4, to 2. Well, 1 and a stop. Okay, so this is usually very detrimental. Now, obviously, if it happens close to the original stop codon, might not be as detrimental, but here you're going from 4 to 1, okay? And then here is where we have an insertion. So we inserted, so we changed the rest of them, Okay, so this usually also big effect. Once again, usually detrimental. And then this last one is a frameshift mutation, so this was an insertion, this was a deletion. Okay, once again, we changed the rest of them. All right, so these usually cause a lot of problems. Okay, see, I told you this was going to be short. All right, here's your wrap up. So once again, I want you to transcribe and translate and create a substitution mutation that changes the fifth T to an A. What kind of mutation is it? All right, and then I want you to find out what kind of mutations causes these diseases: sickle cell, cystic fibrosis, and Tay-Sachs. All right, see you in class. Bye-bye.